Good evening, everyone. Today, I welcome you all to the session. Today, we are conducting 15th session of Good evening, everyone. I welcome you all to this session today. Today, we are conducting 15th session of Pragnavaridhi lecture series. We have today Vaidya Jaibheem Reddy sir. Sir will be teaching about muscle physiology and adipose tissue. Panchajanya, the divine conch of Bhagavan Sri Krishna, which was blown during Kurukshetra, indicating the start of the Dharma Yuddha that followed. There are all the efforts were done for the establishment of Dharma. Dharma was kept over personal relations and motives. Keeping the same motive in mind, our organization will work towards establishing the importance and necessity of following and imbibing the culture, accepting our roots, and making people realize the true essence of Ayurveda, not only as a treatment modality but also as a way of life. All these while walking in the path of Dharma. Hence the motto Shreyam Bharatiyam Arogyam Ayurvediyam. The founders of this organization started this because we felt the need to spread the knowledge of Bharatiya culture, Ayurveda, and its values to all the strata of people in the every corner of the world. Also, the founders being Ayurveda aspirants, know the inside out of today's medical field. Hence, they aim to make people aware of this and also try their best to solve this on the go. In order for all this to be achieved, we have set up various wings with working principles and objectives that help achieve the goals faster and easier in the coming years. The wings we are divided as Team Swami Vivekananda, Team Ashwins, Team Veda Vyasa, Team Sanjaya and Nalanda. Coming back to the members involved in our organization, we have doctors, healthcare workers, teachers, students, and people from almost every single occupation available aiming at working together and bringing forward the true essence of Bharat and her traditions. So, all those who fit under these categories are welcomed with honor and privilege. Today in the session, we have Vaidya Jaibheem sir. Sir will be teaching about muscle physiology and adipose tissue. Sir is currently working as the Associate Professor in Department of Kriya Shareera, Patanjali Bharatiya Ayurvignana Evam Anusandhana Samsthana Haridwar. Sir has completed UG from KLE University, Sri BMK Ayurveda Mahavidyalaya, PG Studies and Research Center, Belagavi, and PG Studies from SDM CH Asan. Sir has pursued NPTEL certificate through course in Educational Leadership by IIT Kharagpur, funded by MHRD Government of India. Sir has also pursued Master Trainer Certificate course in Standardized Prakriti Assessment Scale and Ayur Prakriti web portal conducted by CCRAS New Delhi, Government of India. Sir has delivered guest lectures in different Ayurveda colleges of India. Sir is former Ayurveda consultant and head Herbal Department, Jindal Nature Cures Institute, Bangalore. Sir has published a review article in peer-reviewed journals. Sir presented and paper presentations and poster presentations in national and international seminars. Sir has attended various national and international workshops. Sir has secured first rank to the University RGUHS in Ayurveda Vachaspati, MD Ayurveda in Kriya Shareera examination held in October 2017. Sir, we are very glad to have you in this session today, sir. You can start the session now, sir. Over to you, sir.
Thank you, Miss Neha. Good evening, all. Namaste to all. So we'll start a session by chanting Omkar mantra 3 times. So everyone just close your eyes, take a deep breath. Good evening, all. So Dr. Miss Neha has already introduced me. Myself, Dr. Jaibheem Reddy. I am working as Associate Professor in Patanjali Ayurveda College, Haridwar. So good evening, all. So this session is purely for the students who are going to the forthcoming examination, which will be held at the last of this month or probably the next month in the different parts of different India, right? So the topic has assigned me today is muscle physiology and adipose tissue. Actually, both the divisions, that is muscle physiology and the adipose tissue, physiologically they are entirely different from each other. Their functions, or what to say, their anatomical structures, they are entirely different, but still we will have some correlation between them. So I have divided the entire presentation into three parts. The first part I will be dealing with the anatomy and the basic introduction regarding muscle. Then in the second part, I'll be teaching you the muscle physiology. Then we'll move on to the next topic, that is adipose tissue. So let's begin our journey to understand what is muscle and muscle physiology. Next slide, please. Okay, first, so before going to study muscle in detail, let's understand what is a muscle, right? So everybody will be knowing muscle is a muscle, right? So when you want to define a muscle, it is nothing but a bundle of fibrous tissue in human or animal body which has ability to contract. Of course, the main function of muscle is contraction. What is contraction? Shortening in its length. So whenever any tissue and any element can have a ability to shorten its length, so that is called as contraction. And whenever you can widen it, that is called as elasticity, right? So the bundle of fibrous tissue in the body which has the ability to contract or produce movement or maintaining the position of the body parts, that element to be called as a muscle. So coming to its origin, all the muscles have their origin from the mesoderm, except some muscles like iris, ciliary bodies, they origin from the ectoderm, right? So this is not needed, but still, how exactly muscle has been origin? They have origin from two layers of our tissues. One is from mesoderm, another is from the ectoderm. Next slide, please. So when it comes to the composition of a muscle, so muscle, mainly the skeletal muscles or even cardiac and smooth muscles, they are made up of rich amount of water source. That is, they are made up of 75% of water and 25% of solids. Among solids, the major portion is shared by proteins. That is, around 20% is made up of proteins and 5% the rest of organic and inorganic materials. Together, they form a muscle. So entirely muscle, it is rich in proteins and it is very rich in water content. So just a basic introduction for examination purpose, right? So already I have studied this. Just I will brief it. The classification of a muscle is done based on three strategies. One is whether they are having a presence or absence of striations in them, depending on their control and depending on their locations. So depending on the striations, next slide please. So if a muscles have any cross striations, they are called as striated muscles. Example is skeletal and cardiac muscles, they belong to this category. And non-striated muscles, they do not possess any striations in them. So the examples are smooth muscles. The second classification is depending upon their control. So based on their control, the muscles can be classified as voluntary and involuntary, means which you can control voluntarily. Those muscles are categorized under voluntary muscles. They are skeletal muscles, which are innervated by somatic nerves. And the involuntary muscles are cardiac and smooth muscles, which are innervated by the autonomic nerves. The third classification is based upon their locations, right? Where exactly they are located. Like skeletal muscles, they are adhered to the skeletal system of a body. Smooth muscles, they comprises all the viscera, that is the internal organs. And cardiac muscles, they are specified only to the cardium, that is heart. So based on the locations, they can be classified into skeletal muscles, smooth muscles, and cardiac muscles. So we'll just brief out each groups individually, right? Now coming to skeletal muscles, I will be dealing in detail with the skeletal muscles in the forthcoming slides, right? So these are striated and voluntary in control, and they are made up of 40 to 50% of a body mass. Means they form 40 to 50% of a body mass, and they are anatomically they are long, cylindrical, and unbranched cells, and they are arranged in a parallel lines. They are multinucleated. Next slide, please. So they will be multinucleated and they will be anchored to the bones by the help of tendons, and they can contract very quickly and also undergo fatigue very quickly, right? So right now I will not hook up with what you mean by fatigue. Hope so you people know fatigue. Fatigue is there will be no further contraction. How exactly fatigue happens in the skeletal muscles, we'll see further. And these muscles are supplied by somatic nerves. So this is about the skeletal muscles. I'll be dealing this in detail in the coming slide. Excuse. The next group of muscles is cardiac muscles. So even as skeletal muscles, even cardiac muscles are striated and are involuntary. These muscles, they form the myocardium of a heart. Heart is made up of three layers, and the myocardial layer is made up of these cardiac muscles. And cardiac muscles, they are also cylindrical, short, they are mononucleated, but the difference between skeletal and the cardiac muscles is here the cardiac muscles will be branched and they will be having an intercalated disc in between. So if you can see with the image, so there is a brown color band which is present between the two muscle cells that is called as intercalated disc. These are the two differentiating features from skeletal muscles to the cardiac muscles. That is, one is the presence of branching, second is intercalated disc. Next slide, please. And these cardiac muscles, they will be supplied by autonomic nerve fibers, right? So whereas skeletal muscles, they are supplied by somatic nerve fibers. Here, the cardiac muscles are supplied by autonomic nerve fibers. And what is the importance of branching here is whenever any cardiac muscle is excited, means whenever any cardiac muscle is stimulated, right? So that stimulation will be passed towards the entire set of a cardiac muscles which is present in the heart. So entire cardiac unit will act as only one unit, which is called as functional syncytium, right? So by the help of branching and the presence of intercalated disc, the excitation will move from one cell to the another cell so that all the muscles together, they will act as a single unit. So this aspect is called as functional syncytium. And this can be made possible only by having the branching and intercalated disc, which make a unique muscles from the skeletal muscles. And these skeletal muscles, they have different functions in the heart. So everybody will be knowing the muscles will be having only the property of shortening in its length. But when it comes to cardiac muscles, the same cardiac muscles will act as a muscle unit for pumping action of a heart. Some modified cardiac muscles, they can make pacemaker. That is, the pacemaker is sinoatrial node. Even sinoatrial node is not separate organ. It is just a modified cardiac muscle, right? So the modified cardiac muscle itself makes a pacemaker. Even all the conductive system of a heart, that is the atrioventricular AV node, bundle of A's, Purkinje fibers, or the internodal fibers. All these things are nothing but they are also modified cardiac muscles, right? So the cardiac muscles in the heart, it is having three functions. One is the contractile unit, which helps for the cardiac beat or heartbeat. Second is the modified cardiac muscle itself make the pacemaker. The third is they form a conductive system of a heart. So this is regarding cardiac muscles. Now we'll move on to the smooth muscles. The smooth muscles, as the name suggests, they are not smooth in touch, but they are smooth in appearance because the two form of other muscles, that is the skeletal muscle and the cardiac muscles, they were having cross striations in them, whereas those cross striations are absent in this set of group of muscles. So these are called as smooth muscles, or you can also call them as a plain muscles. And where they are situated, mainly they are situated in the visceral. Also, you can call them as visceral muscles. So how they appear? They appear non-striated. How they are controlled? They are controlled involuntarily, means they are not under your will. So whenever they are not having under your will, this will be supplied by autonomic nerve fibers, right? So anatomically, these are spindle shaped. They are also uninucleated, but whereas skeletal muscles, they are multinucleated. They will be having irregular arrangement of actin and myosin fibers. Usually they are arranged in the layers, but they will be having well-defined myofibrils and the other structures, whereas this well-defined myofibrils and sarcomere will be absent, which are present in the skeletal muscles and cardiac muscles. Next slide, please. So these form the main contractile unit of all the organs together, right? You can just take a screenshot of these things where exactly the smooth muscles are present. No doubt, almost all the body parts have been supplied with the visceral organs will be having smooth muscles in them. So based on the action or shortening property of these muscles, various other actions will be taking place. So just pay concentration on the last point, twist like corkscrew during contraction, right? So you need to understand this concept. What is this concept is, so when it comes to cardiac muscles and the skeletal muscles, they will shorten the length, right? They will shorten the length vertically, right? But whereas when it comes to smooth muscles, they will twist, right? So they will twist like a corkscrew. Whenever you open up the bottle, the way the fashion you open the bottle, in the same way these muscles will twist around themselves so that they will be having shortening in their length. So you can appreciate this in the next slide. Next slide, please. So you can see here on the right hand side, you can see the smooth muscles whenever it is relaxed and contracted. So you can pay attention on the second and the third diagram on my right, on the right hand of the screen. The first muscle shows that whenever the smooth muscle is in relaxed form. So how it get contracted? It contracted like a corkscrew. Whenever you open up the bottle, the way you open the bottle means one thing you will be rotating it anti-clockwise, and the other thing you will be rotating it in clockwise direction. In the same way, the entire spindle, it will rotate in the corkscrew fashion so that it will shorten its length. So this is how exactly the smooth muscle will contract. It will not contract as like skeletal muscle and cardiac muscle. Why? Because in the skeletal muscle and the cardiac muscle, you will be having well-defined actin and myosin filament which are arranged parallelly. So that whenever there is a contraction, they can contract by shortening the length in a vertical fashion. Whereas here, the arrangement of actin or the myosin, they are irregular. So whenever there is a sliding phenomenon, each and every part will slide not in a parallel fashion, but they will slide in a corkscrew fashion so that it will twist around itself to shorten its length. So by doing this, this it will be having different function. Next slide, please. So in the coming slides, I will be explaining what is the function of smooth muscles in different system. For example, in cardiovascular system, except heart, right, except the cardia part, the vascular system is supplied by smooth muscles, right? So in the respiratory system, to regulate the inflow and outflow of the air, digestive system helps in the gut movement, etc., right? Where there is a presence of smooth muscles, they will twist in a corkscrew fashion and they will exhibit different functions. So next slide, please. So the same way they help in the renal system, they help in the reproductive system. Next slide, please. So the same fashion, they will help in the renal system and the reproductive system. For example, in renal system, the smooth muscle regulate the renal blood flow or the glomerular filtration rate. In reproductive system, for ejaculation of the sperm, or in females, whenever the movement of ova in the tract. So all these things are guided only by the help of smooth muscles. I already explained how exactly the smooth muscle will shorten its length. It will be shortening its length with the help of corkscrew-like adjustment, right? So why I'm explaining these things in detail here itself is that in the next part, I will be explaining only regarding the skeletal muscles, which is there for your the academic purpose. But these smooth muscles will be usually excluded in the academic purpose. How exactly it will be contracting, how exactly they will shorten its length, right? So you need to understand the smooth muscle contraction is not as same as skeletal muscle and cardiac muscle, right? To understand skeletal and cardiac muscle, it is very easy. But when it comes to smooth muscle, it makes little bit difficult to understand how exactly it will be contracting. So here are certain differentiating features between skeletal muscle, cardiac muscle, and the smooth muscles, right? So no need to explain here. So whatever I explained before, the same thing has been tabulated here. You can just take a screenshot and it's available in many textbooks. But usually this will be asked in the examination, saying what are the differentiating features between the skeletal muscle, cardiac muscle, or the smooth muscles. Next slide, please. Next slide. So now, so till now we have studied what are the different three types of muscles, how exactly they are different, right? In brief, right? So now we'll be discussing in detail regarding skeletal muscle, right? So which is there for your academic purpose. Now to understand the muscle, I already defined what is a muscle, right? It is an unit which has ability to contract when upon the stimulation, right? So now what is muscle? You understood. Now we'll understand what is muscle mass, right? A muscle mass or a muscle tissue, it is made up of large number of individual muscle cells, or they are also called as myocytes, right? So the myocytes or the muscle cells together, whenever they are present in a compact structure, that entity is called as muscle mass, right? The muscle cells are commonly called as muscle fibers. So there is no any difference between muscle cell and muscle fibers. Why they are called fibers? Because they are elongated and cylindrical in appearance. So they are called as fibers. Skeletal muscles are multinucleated and they are arranged parallel to one another with some connective tissue in between. Means all the cylinders together, the muscle cells, they are arranged parallelly, one beside the other, and they are multinucleated. Next slide, please. So here you can understand what is a muscle mass. The elongated muscle fiber, which is present on your right hand side, that is a single muscle cell, right? So all the group of muscles, they will be present in a group that is called as fasciculars, and all the fascicular together, they make a muscle mass, right? So this is a diagrammatic appearance. The same thing is been explained in the next slide. Next slide, please. So each muscle fiber is covered by a connective tissue layer which is called as endomysium. I said that a single muscle fiber is covered by a connective tissue layer which is called as endomysium. In the muscle, all the muscle fibers together, they are arranged in a group which is called as fasciculi. And all the fasciculi together, each fasciculus is covered by perimysium, and all the fasciculus together is covered by one more connective tissue sheath which is called as epimysium. And this entire group of fasciculus together, which is covered by epimysium itself, is called as muscle mass. So here you can see a cross-sectional diagram. So you can see an individual muscle fiber which is covered by a, it is not visible here, which means the individual muscle fiber which is covered by epimysium, and all the group of muscle fibers together, they are called as fasciculars, and all the fasciculars together, they make up of muscle mass. Now we'll study what is individual muscle fiber or a muscle cell. I already told what is muscle fiber or a muscle cell. They are one the same because they are present. They appear like a cylindrical in appearance, so they are called as muscle fiber. Now, each muscle cell or a fiber is cylindrical in shape, and the average length varies between 3 centimeter, or it can vary between 1 to 4 centimeter, but average you can consider as 3 centimeter. All the muscle fibers are attached to a tough cord of connective tissue called as tendon. We are dealing regarding skeletal muscles, right? So all the muscle fiber, they will be anchored to the bone by via means of tendon. You can see a tendon is a tough cord of connective tissue. All the muscle tissue ends there, and from the help of tendon, they are anchored to the bone. Each muscle fiber got one or more nuclei. Already we have studied the skeletal muscles are multinucleated. All the organelles of a muscle fiber have a same function as those of other cells, right? For example, coming to the next slide, the same entities which are present in a cell, what you have studied before, the same entity is also present in the muscle cell with some change in the name pertaining to the muscle. For example, the plasma membrane in the muscle cell is called as sarcolemma. The cytoplasm is called as sarcoplasm. And endoplasmic reticulum is called as sarcoplasmic reticulum. And the hemoglobin which is present in the other cells like RBC is the same thing. You will be having myoglobin in the muscle cells. Means all the entities which you study in a human cell, the same entities is also present in the muscle with a different name, right? So coming to what is individual myofibril, right? So now here the most of the students, they will get confused to understand what is a muscle fiber and myofibril, right? Muscle cell is the individual cell, right? Within that individual cell, you will be having multiple myofibrils, right? I think that myofibril is one of the cell organelle which is present in the muscle cell. But the thing is, the myofibril is present all along the length of a muscle cell. As you can see, other cell organelles like the Golgi apparatus, mitochondria, they are specified with a specified place. But when it comes to myofibril, it is present all along the length of a muscle cell. What is the length of a muscle cell? That will be the length of myofibrils, right? So don't get confused with muscle fiber and myofibrils. Myofibril, all the muscle cell. Previous slide, please. Previous slide. Yeah, fine. So the difference between myofibril and the muscle fiber is a muscle cell and myofibril is a individual unit which is present within a muscle cell, right? So it lengths all along the length of a muscle, and as the length of a muscle, there will be a length of myofibril. Coming to the next slide, the same diagram is been depicted in a pictorial form here. So you can see first there will be a muscle. In the muscle, how muscle is made up of? It is made up of group of vesicles. What is vesicle? Group of muscle fiber. So in individual muscle fiber, you will be having myofibrils. Next slide. So when you study a microscopic structure of a myofibril, right? So we are entering into the deeper aspect of a muscle cell, right? We are studying a individual unit which is present in a muscle cell, that is a myofibril. So whenever you study this myofibril under a microscope, it reveals a two alternating bands in a myofibril, right? So these alternating bands are formed by actin and myosin pigments, right? All the proteins which are present in the muscles. So the two bands are I band and A band. Next slide. So what is I band? I band is called as isotropic band because whenever a polarized beam of polarized light is focused on that band, it reflects in the same angle. So it is called as isotropic. And dark band is called as A band. Why it is because whenever a polarized light is focused on that area, it is refracted at different directions, not in the same direction. So it is called as anisotropic band. So based on their projections of a light, so they have been named as I band and A band. So light band will be I band and dark band will be A band. So next slide, please. So here you can see in the picture what is A band and what is I band, right? Which is present in the red color, that is called as A band, and which is having light color, that is called as I band. So in intact muscle fibers, I band and A band of adjacent myofibrils are placed side by side. What does it mean is, in an individual muscle cell, you will be having n number of myofibrils. So how exactly they are placed? The A band of one myofibril is exactly present on the same line in the A band of its adjacent myofibrils. They are present exactly in the same fashion with the same distinction. So whenever you see an individual muscle, even the individual muscle will appear with the dark I band and A band, right? Why? Because they are perfectly positioned in the same line. So this characteristic appearance of dark band and the light band, it makes the cross striations in the muscle cell or muscle fiber. So in between the two portions, that is in between the two in between the two portion, it is divided by a Z line. So you can see in between two red lines, there is a light band, and in between the light band, you can see one more line which is called as Z line, right? Means the two units, whenever they join together, that area is called as Z line, right? The portion between the two Z line is called as sarcomere. So what is a sarcomere? The portion which is present between two Z lines. What is Z line? The Z line is an area where the two units intersect, right? Coming to the next slide. So next slide, please. Here you can see between the two Z lines. You can see in the picture like Z disc, right? Previous slide, please. You can see as a Z disc. So in between this Z disc, you will be having a portion of sarcomere. So what is sarcomere? In the sarcomere, you will be having two dark band and one light band, right? So this together it forms what to say the sarcomere. So what is sarcomere? Next slide, please. So sarcomere is defined as structural and functional unit of a skeletal muscle. It is also called the basic contractile unit of a muscle, right? So now initially you studied what is a muscle, then you studied what is muscle fiber. In the muscle fiber, you studied what is myofibrils. In the myofibrils, you are studying sarcomere, right? So you are going into the deeper aspects now. So what is sarcomere? A portion present between two Z lines is called sarcomere. So how you can define it? It is a structural and functional unit of a skeletal muscle, or this is the basic contractile unit. Whatever the contraction takes place, it takes place in this unit. To understand how exactly the muscle shorten in its length, it's via the shortening the sarcomere. And each sarcomere extends between two Z lines of a myofibrils. The next slide, please. So now here the sarcomere is been drawn pictorially, right? The same thing what you have studied there. Now here is a pictorial diagram of a sarcomere. The two lines that is which are a light blue, a dark blue in color, they are called as Z line, right? And light blue color they are called as I band, which is light band. And in between, wherever you see the red lines or between the portion that is called as A band. So how sarcomere is made? It is made up of two I bands and one A band, right? So this is sarcomere. So now in the middle of A band, there is a light area which is called as H zone. So there is no darker area. You can see in the picture wherever there is an A band, in between you will be having a H zone. In the center of that H zone, you will be having a line which is called as M line, right? So what is this M line? From this M line, uh, the proteins will be having taking its storage. That is, the myosin filament will be attached in between by the M lines, and actin film will be attached to the Z line. So to remember the sarcomere, myosin will be attached to the M line, which is at the center of sarcomere, and actin filament are attached to the two Z lines which are present on the either side. So this entire unit between means two portion of actin filaments and one portion of myosin filament together they form sarcomere, which is the contractile unit of a muscle. Next slide, please. So now the electronic microscopic study of a sarcomere, it says there are two types of filaments. One is actin filaments and myosin filament. Actin filaments are thin filaments which origin from the Z line, and they are having diameter of 20 angstroms and the length of 1 microns, right? These extend from either side of a Z lines. So these actin filament, they are made up of three type of proteins. So the entire actin filaments which get origin from the Z line, they are made up of three type of proteins. In the image below, you can appreciate one molecule is made up of light blue color, another is totally purple, and one is in the pink color, right? So the purple color line, which is having a crisscross fashion like DNA, that is made up of tropomyosin. And the light blue color bubbles, they are called as actin. And the pink color big bubble, what you appreciate in the image, that is called as troponin, right? So this entire unit is called as actin filament, right? So these actin filaments are in turn made up of three type of protein. One is actin, tropomyosin, and troponin. Next slide, please. So when it comes to myosin, it is made up of only one type of molecules, but it is having two different portions. One is the tail portion and the head portion. The elongated portion which origin from the center line is called as head, and whenever head is attached to the center line, that is called as tail portion. And here you will be having one myosin binding site. So you can see this molecular diagram, the same diagram what you studied before, the sarcomere, the two Z lines. From the Z lines, there will be actin filament in the middle, that is called as M line. From the M line, you will be having myosin filament. So one more aspect you need to study in the anatomy of a muscle, that is sarcotubular system. So what is the sarcotubular system? The sarcotubular system is a network of tubules, right? Sarcotubular system. What is sarco? Sarco is a name for sarcomere, right? A group of muscle or a division of a muscle. And tubular system. The tubular system which is present within the sarcomere, that is called a sarcotubular system. And this sarcotubular system is made up of two tubules. One is T tubules and L tubules, right? The T tubules, they pass all along the length of a muscle in a cross section form, from one end of a muscle to the another end of a muscle. They are passing from one end to the another end. What is this L tubules? L tubules are nothing but they are the endoplasmic reticulum. Previous slide, please. And the L tubules are nothing but the endoplasmic reticulum of a muscle cell is called as sarcoplasmic reticulum, or that is called as L tubules. The main thing you have to understand here is these L tubules will be having calcium in them. So what is the function of calcium? We'll study further. But for instance, you need to understand what is the function of L tubules. L tubules store calcium. What is the function of T tubules? T tubules will be passing the what the electrical changes or action potential from one end of a muscle to the another end. Next slide, please. So now till now we have studied basic anatomical part of a muscle, right? So just to have a refresh, each muscle or muscle mass is made up of group of muscles, right? What is a muscle cell? Muscle fiber, or they are called as a muscle fiber or myocyte. Within the muscle cell, you will be having myofibrils, right? They are a unit of a muscle cell. In the muscle, you will be having number of myofibrils. How myofibrils are formed? Myofibrils are formed by a group of sarcomere. What is sarcomere? A group of actin and myosin filament which is having two things that is made up of a sarcomere between two Z lines. So what is sarcotubular system? A network of tubules. It is called a sarcotubular system. Now having with this basic anatomical features, now we'll go to the next part of this presentation. That is how exactly muscle will contract. Next slide, please. Next slide. So now when the muscle will get contracted, the muscle will contract whenever it is stimulated. It should have some stimulation, right? No doubt the stimulation may be by the will if you want to lift an hand, but the stimulation has to be conveyed from the center to the periphery, that is to the muscle. So whenever a muscle contract, whenever it is having a particular stimulus, then only a muscle will get contracted. So now what is contraction? It is a physical or mechanical event, right? What is contraction to define? Whenever something is shortening in its length, that is called as contraction, right? So in addition, several other changes also occur in the muscles. Now we'll study what are the other changes which are taking place in a muscle. So just for instance, muscle will contract whenever it gets stimulated and they're having different changes. Now what are the changes? The first is electrical changes. That is utmost important, right? So whenever a muscle contract, there are mainly two events. One is the electrical event, second is the mechanical event. Always the mechanical event is preceded by electrical event. Means first you need to have an electrical event, then it is followed by mechanical event. The first changes is electrical changes, second is physical changes, then histological, then chemical and thermal changes. Now we'll study one by one. What is the physical changes which is here? I have not explained what is the electrical changes. Electrical changes here is the change in action potential. Whenever an action potential reaches the neuromuscular junction, means a junction between the nerve and the muscle tissue, that portion is called as neuromuscular junction. So whenever action potential reaches the neuromuscular junction, the potential difference at the cell membrane with the myofibril level, it changes. That is called electrical changes. After that electrical changes, there will be physical changes. What is a physical change? Change in the length of a muscle fiber or change in the tension. Not only the change in length, even the change in tension of a muscle is also called as contraction. So this contraction may be of two types. One is isotonic and isometric, right? So how need to understand? Isotonic means if there is no change in the tension, if there is only change in the length, that is called as isotonic contraction. If there is no change in the length, only change in the tension, that is called as isometric contraction. These words you will encounter whenever you study cardiac cycle, right? For example, whenever you study cardiac cycle, then you'll understand what is isovolumetric contraction or isometric contraction. What does it mean? That you have a muscle, right? For example, think that you have a muscle of 4 centimeters. If that 4 centimeter is changing its length to 3 centimeters, then it is changing its length. Without changing the tone, it is just changing the length, that is called as isotonic contraction. If the same muscle of 4 centimeter, without changing the length, it is remaining in its original 4 centimeter, but the tension is developed within the muscle, that means there is a change in tone, but there is no change in length. That kind of contraction is called as isometric contraction, right? So now you can understand what is isotonic and isometric. Just focus the word. Isotonic means there should be same tone. Iso means same, tonic means same tone. Next, isometric means same and metric means length. If muscle length is not altered, only tone is altered, that is called as isometric. If muscle length is altered, not tone is altered, that is called as isotonic. Coming to the next, histological changes. So in histological changes, what happens is, you studied sarcomere, right? For example, if a length of a sarcomere is 4 centimeter, just for instance, to make you understand, It will not be 4 centimeter, right? So the length of a sarcomere, whenever it shortens, the two Z lines come in closer way, right? So what is sarcomere? A distance between two Z line or the portion present between two Z line is called sarcomere. Whenever there is a muscle contraction, these two Z lines they come in close proximity. So there is change in length. So these kind of changes are called as histological changes because there is change in their length. And how exactly they will contract? Because of actin filament gliding over the myosin filament. So we'll understand in the next slide. Next slide, please. So here you can appreciate before contraction, after contraction. You have two images, image A and image B. So you have having the same sarcomere. What is sarcomere? A distance, the portion between two Z lines. The blue color thick lines which are present vertically, they are Z line. From the Z line, the another blue color which are placed horizontally, they are called as actin filament. And the red color, they are called myosin filament. So whenever there is contraction, the two Z line comes in close proximity. These are some of the histological changes which is happening in the sarcomere. Coming to the next slide. So now what are the molecular changes happening there? So the molecular mechanism is responsible for formation of actomyosin complex. So this is the main phenomena you need to understand in muscle physiology, right? So means how exactly there is shortening of a sarcomere or how exactly there is shortening of a length in a muscle, which is called as contraction. So this process, it includes three stages. Stage one is excitation contraction coupling. Second thing is the role of troponin and tropomyosin. And the third is sliding mechanism. Now we'll study these three events one after other. Right? The first is excitation contraction coupling. Next slide, please. So this is the process that occurs between the excitation and contraction of a muscle. So what is excitation? Excitation is a type of stimulus. So what is a stimulus here? The stimulus is nothing but a change in action potential which is carried all along the length of a nerve which is reaching the neuromuscular junction. So whenever an action potential reaches the neuromuscular junction, from that time to the shortening of a length of a muscle, this period is called as excitation contraction coupling. From excitation to the contraction. And this involves a series of activities which are responsible for the contraction of a excited muscles. Now, what are those series? We'll study in the next slide, please. Next slide. Next slide. Next slide. Sorry, previous slide. Previous. Okay, there is some glitch in the PPT, right? So here I had a schematic diagram of excitation contraction for no doubt. I'll just explain it verbally. So an, whenever a nerve impulse pass or whenever a nerve impulse reaches the neuromuscular junction, the action potential is generated at that part, right? Now this action potential is passing through the T tubules. So you can just remember you are having two types of tubular network. One is T tubules and L tubules. And T tubules pass all along the length of a muscle fiber. Now action potential reach the muscle. That action potential travels to the T tubules. Then it will reach the L tubules. As I told in the L tubules, what will be stored? There will be storage of calcium ions, right? So whenever this action potential reaches the L tubules, there will be release of calcium. Now this released calcium will bind with actin filament and that will produce contraction. Means whenever a calcium ions are released, whenever they bind with troponin of the calcium binding site of a troponin, so by that there will be binding of actin and myosin and there will be sliding theory. So now you can understand here is calcium ions, they form the basis of excitation contraction coupling, right? So if there is no release of calcium, there is no muscle contraction. So what exactly happens in excitation contraction coupling is whenever there is excitation, what is excitation? Whenever a nerve impulse reaches the neuromuscular junction, from that part to the shortening of a length, whatever events take place, that is called as excitation contraction coupling. So what happens? Action potential reaches the cell membrane of a muscle cell. That action potential from the membrane, it is traversed through T tubules. From the T tubules, it will reach the L tubules. From the L tubules, there will be release of calcium ions and there will be muscle contraction. So this entire period is called as excitation contraction coupling, right? So the image which is been displayed in the screen, previous slide please. So here you can understand the T tubules are the transverse tubules. From their action potential will be moving in, and this makes the release of calcium. So whenever the calcium is released, it will bind with the troponin so that there will be excitation contraction coupling. So what exactly happens is, coming to the next slide. Next slide, please. So now here comes the second stage of muscle contraction, that is role of troponin and tropomyosin. The first was excitation contraction coupling. To understand excitation contraction coupling is simple. There will be release of calcium ions within the muscle cell. So whenever there is a release of calcium ions, what exactly it will make? So normally the head of myosin molecules has strong tendency to get binded with the active site of actin. But in a relaxed stage, this is covered by tropomyosin, right? So what is tropomyosin? You can see a yellow color filament which is running all along the length of a line, that is called as tropomyosin. So that will be binding the active site. So how exactly it can be removed is by the help of calcium ions. So whenever calcium is released, it will twist the tropomyosin so that the active site or the binding site will be opened so that it can bind with the myosin filament. So the next slide, please. Next slide. So here you can understand whenever a muscle is relaxed, you can see the actin filament which is in blue color and tropomyosin it is in purple color and troponin complex is present in pink color, right? So actin binding site, you can see a black color dot that are called as actin binding site. You can see in the first image all the binding site is covered by tropomyosin, means no binding site is free. So if there is no binding with actin and myosin, means there is no sliding phenomena. So what is the role of calcium here? So whenever calcium is released, it will twist the tropomyosin component so that the binding site are opened. You can appreciate that in the second picture, right? In the first image, all the binding site was closed by the help of tropomyosin. Whenever calcium is released in excitation contraction coupling, that will open up the binding site so that it will be free for binding with the myosin filament. So whenever this happens, there will be sliding phenomena. That is the next step in contraction. Next slide, please. So this is the final step. First was excitation contraction coupling, means there will be release of calcium. Second is role of troponin and tropomyosin. So what is role of troponin and tropomyosin means whenever calcium releases, there will be twisting up of tropomyosin so that there will be opening up of binding site. So whenever binding site is opened, that will bind with myosin filament and there will be sliding phenomena. So this is the third phenomena which is happening in the muscle contraction, that is sliding phenomena, right? That is called as ratchet theory or walk along theory. Each cross bridges from the myosin filament, they will get attached with actin and that actin will pull backwards so that there will be contraction, right? It is as same as whenever you intersect two different fingers of your hand between one after the other. So you just hold two hands together. You just intersect each fingers in the gap between the other fingers of the opposite hand. So you can just move to and fro. How exactly they will move between two fingers? The same thing will be happening here. The myosin and actin filament, whenever they bind, they will pull apart to the M line. Next slide, please. After binding with active site, the myosin head is tilted so that the binding site is open, and this tilting will create a power stroke. So you are having two images, the first image and the second image. That in the stage one, it will bind with an myosin binding site. The actin will bind with myosin head. So this myosin head will give a stroke towards the M line, means towards the center of a sarcomere. So whenever this happens on the either side, from the left side I band and the right side I band, they will be moving towards the center. So there will be shortening of a sarcomere. So so by this way, there will be shortening in the length. So what happens in the muscle contraction is, one is excitation contraction coupling. There will be release of calcium. Whenever calcium is released, troponin and tropomyosin comes into an action. They will open the binding site. The third stage is whenever binding site is open, the myosin will bind with actin, and this myosin will pull the actin filaments towards the center of the sarcomere. So when this is happening, there will be shortening up of a sarcomere, which is called as a muscle contraction. So these are the three changes which happens during a muscle contraction. Next slide. The same thing. Next slide. Next slide. Previous one. So here overall, what are the changes of a sarcomere? One is the length of sarcomere decreases as Z lines come close to each other. Second thing is length of I band will be decreased. H zone will be either decreased or totally disappears, but whereas A band remains the same. Next slide, you can understand here. Next slide. So you can see in the previous image what is the length of I band and what is the length of A band, right? So whenever there is a muscle contraction, there is a change in the length of I band, but A band remains the same because the myosin filament, which is present intact, they will move the actin filaments towards the center aspect. So this is shortening in its length. So whenever a muscle is contracted, means whenever a sarcomere is intact. So now whenever there is a muscle contraction, now we'll see what happens during relaxation of a muscle. So in contraction, the calcium ions is released so that binding site is opened and there will be having sliding phenomena. So whenever a muscle is relaxed, means how exactly the muscle will be relaxed? The calcium ions will be pumped back to the L tubules. So whatever the calcium ions were released from the L tubules, that is sarcoplasmic reticulum, they will be taken back to the sarcoplasmic reticulum. So whenever the calcium enters back, again the binding site will be closed. So whenever a binding site is closed, so there will be detachment of myosin. So whenever there is a detachment of myosin, they will come back to their original place. Means both the I bands will move apart because they are elastic tissues, right? So this happens. There will be a relaxation of a muscle, right? Coming to the next slide. If possible, you can play this video. Fine. I think it's not supported. There was an entire video showing that how exactly the muscle contraction will take place. So with the technical glitch, it's not possible to play a video here, right? So now we have completed two parts of our presentation today. One is the anatomical part of a muscle. Second is the physiological way how exactly there will be contraction of a muscle. Hope so you understood the concept. If you have any problem in understanding, you can directly contact me or the organizer. By the thing, they will be directing your queries to me, then I can answer your queries, right? So now we'll move on to the next part of today's presentation, that is adipose tissue. So actually, adipose tissue was a topic which was assigned to me. Even I included lipoproteins because you need to study them end to end. So I included adipose tissue and lipoproteins here together. Next slide, please. So what is adipose tissue? So adipose tissue is a fat or loose connective tissue that forms the storage site of a fat in the form of triglycerides. So whenever a triglyceride is stored in some particular area, that itself forms the adipose tissue. It is composed of adipocytes, right? Means the cells of adipose tissue, they are called as adipocytes, or they are also called as lipocytes or fat cells. So when you call a person as obese, it is not the content or it is not his body weight, it should be the content of fat which is present in them. So nowadays BMI is outdated. So what is BMI? How you calculate BMI? You will calculate BMI by weight divided by height in meter square, right? But whenever it comes to a body weight is not particularly made up of fat. The body weight, 50% of body weight, 40 to 50% is made up of muscle itself. Then remaining it will be made up of bones. So whenever a person is too heavy or too FT, it doesn't mean that he is having only fat tissue. It means that he may have even muscle tissue or he may having good amount of bone tissue or the skeletal tissue. But to distinguish between the obese person, there is one more technique which is called as electrical impedance. So by that you will calculate what is the fat percentage in the body. Then you can designate a person as obese or not obese, right? Next slide, please. So there are two types of adipose tissue in the body, right? One is white adipose tissue, another is the brown adipose tissue. So what is white adipose tissue? Next slide. So white adipose tissue is distributed throughout the body, which is present beneath the skin, or it is called as subcutaneous fat, right? Which is present below the skin. That is called as white adipose tissue, and it is called as subcutaneous fat. It also surrounds the internal organs of a body, right? And mainly it is made up of fat cells which are unilocular, means they contain only one large vacuole filled with fat. So in white adipose tissue, you will be having only one single large vacuole which is filled with fat. So what is the function of white adipose tissue? Next slide, please. So white adipose tissue serves three functions in the body. One, it serves as a storage of energy. Whenever there is depletion of carbohydrates, whenever there is depletion of proteins, this adipose tissue will take part in production of energy. Right? That is a ready reckoner. After there is extinguishing of carbohydrates or the glucose, then the body will utilize fat. The next is they act as heat insulator, means they protect the body from the severe cold. So they are called as heat insulation. Third thing is, as they are surrounding the internal organs, they can also act as a cushion, like so that they also protect the internal organs. So what is the main function of white adipose tissue? Is there are three functions. One is storage of energy, second thing is heat insulation, and third thing is protection of internal organs. Coming to the next form of adipose tissue, that is brown adipose tissue, right? Or it is also called as brown fat. So why it is called brown fat? Because of its appearance, right? The brown adipose tissue is specialized form of adipose tissue having function opposite to that of white adipose tissue. Actually, it is not exactly opposite. It is other than the function which is given by white adipose tissue. It is present only in certain areas of a body, such as back of a neck and intrascapular region, and it is abundant in the infants rather than males, right? After infancy, the brown adipose tissue will disappear gradually, and only 1% of total fat present in the body will be made up of brown adipose tissue. It means that in adult life, there will be no brown adipose tissue which is present muchly in the human body, right? Coming to its anatomical structure, next slide please. It is formed by fat cells which are multilocular inversion, means they are having many small vacuoles filled with fat, whereas white adipose tissue, they are having only single vacuole which is filled with fat. Here they are having multiple small vacuoles within them. And why it is called brown adipose tissue is because of high vascularization. There is large number of iron rich mitochondria in these things. So this iron rich mitochondria will give a characteristic dark brown color appearance. So it is called as brown adipose tissue. Next slide please. The function of brown adipose tissue. So when it comes to white adipose tissue, it was serving three functions. One is storage of energy, heat insulation, and protection of internal organs. Here, this brown adipose tissue will not store any lipids, but it generates only heat, right? How? By burning the lipids. So in infants and hibernating animals like frog, etc., they will be having rich amount of brown adipose tissue. So what is the importance of brown adipose tissue is, whenever the body temperature falls below the normal limit, so this brown adipose tissue will burn the fat to generate heat. This is called as non-shivering thermogenesis. So you need to understand this word. What is non-shivering thermogenesis? I know everybody know what is shivering, right? So whenever there is a cold climate outside or cold weather outside, you start feel shivering. So why you feel shiver is because to generate the heat. What is shivering? It is uncontrolled contraction, rhythmic contraction of a skeletal muscles. By that, there will be generation of a heat. That is called as shivering thermogenesis, right? So here, brown adipose tissue without shivering, by just burning the fat, it will produce heat. So this is not very much uh, extent which is present in a human body. It is mainly present in hibernating animals, even in small infant. So what is the function of brown adipose tissue is production of heat, that is non-shivering thermogenesis. So this is regarding adipose tissue, right? There is nothing much to understand in adipose tissue. Just you need to know what are the types of adipose tissue. You will be having white adipose tissue and brown adipose tissue. You have to understand what are their different functions. So now moving on to the next topic, that is lipoproteins. So what are lipoproteins? These are small particles in the blood, right? So lipoproteins are present only within the blood. How they are made up of? They are made up of cholesterol, phospholipids, triglycerides, and proteins. So all these entities together, they form lipoproteins, right? What is lipoprotein? It is a conjugated protein conjugated with lipid elements. It is called as lipoprotein. I hope you can understand what you mean by conjugated proteins, right? These are different types of proteins: simple protein, conjugated protein, etc. Conjugated means whenever a protein is conjugated with non-protein part, that is called as conjugated protein. Here, lipoprotein is a conjugated protein which is conjugated with lipids, right? So what are those lipid molecules? One is cholesterol, phospholipids, triglycerides, and proteins. Now, proteins are beta globulins, which are made up of apoproteins, and these serves as a lipid transporters in the blood. So what is the function of lipoproteins? Transportation of lipids. So this lipoproteins is classified into four different forms. Coming to the next slide. So what are the four different types of lipoproteins? VLDL, very low density lipoprotein, means why they are called low density lipoprotein is because they are having high concentration of triglycerides, moderate concentration of cholesterol and phospholipids. There is no protein in them, and density of lipoprotein is based upon the presence of protein in them. How much protein is present, that much density will the lipoproteins will have. Very low density lipoproteins, there doesn't have much of protein part in them, right? The next is intermediate density lipoprotein, which is called as IDL. So how this is formed? By removing the portion of triglycerides from VLDL. So it forms IDL. Next classification is low density lipoproteins. It is made up of complete removal of triglycerides, right? In VLDL, it was having rich amount of triglyceride. In IDL, some of the amount of triglycerides it's removed. In LDL, there will be total removal of triglyceride, but they will be having some amount of lipoproteins. Like these lipoproteins will be having little amount of cholesterol and phospholipids. Now coming to high density lipoprotein, this high density lipoprotein is made up of rich amount of protein and only low concentration of cholesterol and phospholipids. As I told, the density of lipoprotein is based on the content of protein which is present in them. If you want to designate them as high density lipoprotein, means they should have rich amount of protein in them. Very low density lipoprotein, they will be having only small amount of protein content in them. So now you already you will be seeing in many ads, etc., right? Which is good for body, that is HDL will be good for body. Why? You can understand this in the next slide by seeing the functions of lipoproteins. Next slide, please. So I said the primary function of lipoprotein is to transport lipids via blood. So these are basically they are lipid transporters. So how exactly VLDL function is? VLDL transport triglycerides from liver to different adipose tissue. From liver to the adipose tissue which are scattered all along the body. This will move from livers to the different tissues, right? Like heart, etc. So VLDL is called as bad cholesterol because it is taking fat from the liver and it is depositing it in different aspects of the body. When it comes to intermediate density lipoprotein, this will also transport triglyceride, cholesterol, phospholipids from liver to peripheral tissue. The same thing, VLDL, IDL, they will serve the same purpose from liver to the different parts of the body. Comes to low density lipoprotein, so this will transport cholesterol and phospholipids from liver to organs like heart, right? So LDL is called as bad cholesterol because it is taking fat from liver to the heart, so that there will be atherosclerosis or some other disease happening there, and even there will be myocardial infarction because of deposition of this fat in the coronary artery. So because of these things may happen if there is more deposition of cholesterol. So this is called as bad cholesterol, right? When it comes to HDL, it will transport cholesterol and phospholipids from different tissue back to the heart. Means from heart to back to the liver. It is taking whatever the fat is deposited in the blood vessels back to the liver. So HDL is called as good cholesterol because of its function, right? Basically, all these serve as a lipid transporters. All these are needed by the body. It doesn't mean that you don't need LDL. No, you need LDL to some extent so that it serves the purpose, right? It should transport some of the what to say lipids from liver to the different parts of the body in case of production of energy. But whenever this increases, then the normal limit, there comes the pathology, right? So with these things, I wind up today's presentation. If you have any queries, right, you can comment in the comment box, or else you can send your queries to the organizer. In turn, they will revert it me back, and I can extend whatever help I can do. Right? Thank you. Over to you, Sneha.
Yes, sir. Yes, sir. Thank you, sir. Now we are coming to the end of the session. Sir, we are very glad to have you in this session today. On behalf of our Panchajanya organization, we are very thankful to you for accepting our invitation to conduct this session today. This session will definitely useful for the students who are preparing for the upcoming exams, sir. Once again, thank you so much, sir. We'll wind up the session now.
Thank you, Sneha.
Thank you, sir.